Hi, my name is Sohail. Today I am going to discuss the part 3 of prokaryotic transcription. It includes prokaryotic elongation, prokaryotic termination and hairpin loop. The transcription elongation phase begins with the release of sigma subunit from the polymerase. The dissociation of sigma subunit allows the core enzyme to proceed along the DNA template, synthesizing mRNA molecule in the direction of 5' to 3' at the rate of approximately 40 nucleotides per second. As elongation proceeds, the DNA is continuously unwound ahead of core enzyme and rewound behind it. The base pairing between the DNA template strand and mRNA is not stable enough to maintain the stability of mRNA synthesis. So, RNA polymerase enzyme that acts as a stable linker between the DNA template strand and nascent RNA strand that ensures that elongation is not interrupted prematurely. Once the gene is transcribed, the prokaryotic polymerase needs to be instructed to dissociate from the DNA template strand and liberate the messenger RNA molecule. Depending on the gene being transcribed, there are two kinds of termination signals. One is protein based and second is RNA based. Protein based is also known as row dependent termination, and RNA based is also known as row independent termination or intrinsic termination. It is a process carried out in prokaryotic organisms as a mechanism for stopping transcription. Row dependent termination is controlled by a protein. That protein is called row protein, and this protein tracks along behind the RNA polymerase. As we can see, this is RNA polymerase enzyme, and behind this RNA polymerase enzyme, this is the row protein, and this is the template strand of DNA, and this is non-template strand, and here is the messenger RNA which is synthesized in the direction of 5' to 3'. So, near the end of the gene, polymerase encounters a sequence of guanine nucleotides on the DNA template strand and it stalls to stop. As a result, the row protein collides with RNA polymerase enzyme, and this interaction with the row releases the mRNA molecule from the transcription bubble. Second type of termination, which is known as row independent termination, that is also known as intrinsic termination. It is controlled by specific sequences in the DNA template strand. As the polymerase enzyme nears the end of the gene being transcribed, it encounters a region rich in cytosine guanine nucleotides in the messenger RNA. So, the messenger RNA that is rich with cytosine guanine nucleotides that folds back on itself, and complementary cytosine guanine nucleotides, they bind together to form GC rich hairpin loop. The result is a stable hairpin that causes the polymerase enzyme to stall as soon as it begins to transcribe a region that is rich with adenine nucleotides on DNA template strand. The complementary uracil adenine region of messenger RNA transcript, it forms a weak interaction with the DNA template strand. So, it is coupled with the stalled polymerase, induces enough instability for the core enzyme to break away and liberate the new messenger RNA molecule. Multiple polymerases can transcribe a single bacterial gene, while numerous ribosomes, they translate the messenger RNA into polypeptides. In this way, specific proteins can rapidly reach to a high concentration in the bacterial cell. To sum up, transcription elongation phase that begins with the release of sigma subunit, and the core enzyme, it synthesizes messenger RNA molecule in the direction of 5' to 3'. As elongation proceeds, the DNA is continuously unwound ahead of core enzyme and rewound behind it. RNA polymerase is a stable linker between the DNA template an mRNA molecule. After gene transcription, there is termination signal, and there are two kinds of termination signals. One is protein based, that is known as row dependent termination, and second is RNA based, which is called row independent termination. It is also known as intrinsic termination. In row dependent process, row protein locates and binds the signal sequence in the messenger RNA molecule and signals for cleavage. Intrinsic termination does not require a special protein signal for termination, and it is controlled by specific sequences in messenger RNA. When the termination process begins, mRNA molecule, it forms a stable secondary structure that is called hairpin loop. This hairpin loop is followed by multiple uracil nucleotides, and the bonds between uracil and adenine nucleotides are very weak, and the hairpin loop structure that causes the RNA polymerase to temporarily stall. The pausing of polymerase coincides with the transcription of polyuracil nucleotides. The weak adenine uracil bonds, it lowers the energy of destabilization for RNA DNA duplex, and it allows to unwind and dissociate from the RNA polymerase.